Just to quickly introduce myself, I'm Rodolphe Rougerie from the Natural History Museum in Paris. I'm an assistant professor there and curator for the collections of Lepidoptera. So Lepidoptera is my group of interest with a special focus on some families of big moths, saturnids and sphingids. And I've been involved in a number of barcoding projects with Lepidoptera campaigns when I was working here as a postdoc for 4 years in Guelph. Also on DNA barcoding of type specimen, so degraded DNA, insect community surveys using meta-barcoding and barcoding, soil biodiversity surveys, and now a bit more looking into high-throughput protocol for trying also to decrease the cost of generating barcode for libraries. So in terms of the coverage for France, that's about 50,000 sequences for France and 8,000 named species, as you can see. Then the correspondence between name and bean is relatively equivalent. Like the taxonomy, of course, in Europe is relatively advanced and achieved. It's about a tenth of the plant species that have been barcoded in France. Tenth of the species. Half of the vertebrates. A number of lots of freshwater fish have been databased. And of course, in arthropods, it's still very incomplete. Also, a number of these arthropod species are probably been barcoding already from other neighboring countries in Europe. I wanted to mention also that France has overseas territories, and that hosts actually a lot more diversity than we have in France. I could not get the detail of the number of species from board and number of beans, but that's almost as many sequences that have been produced from these territories. A lot of them from French Guiana, where there's probably the highest piece of Amazonia, so the highest diversity for France. And also, actually, marine life has been barcoded relatively heavily at some point. So marine fish and mollusks from Polynesia, French Polynesia, and New Caledonia. But for the barcode network, we don't have a national network in France. It doesn't exist. But I mentioned a number of structures that have been supporting some work in relation to barcoding in the past, and also at the moment, a few months ago, where was created the Natural Heritage Service, that is a service unit that is governed by the French Agency for Biodiversity, the Natural History Museum, and the CNRS, and that is going to regroup the initiative of the National Pool for Biodiversity Data, which is the structure that supports the participation of France to iBOL 2, and the National Inventory of Natural Heritage. There's a thematic research group on environmental genetics that exists still since 2011 that has been quite active in promoting environmental genetics, genomics in Europe, organizing a conference each year, and also helping for student and researcher mobility. There's an RCIS network that has been focusing on molecular characterization of species of agronomic interest. It hasn't been very active lately. It's something that was more active, I guess, several years ago, but it still exists. And my last point here is the Entomosant project that is led by Carlos Lopez-Varamonde from the National Research on Institute of Agronomy. And it's actually not yet funded, but the chances are relatively good. And they want to make it's a regional center that wants to regroup entomological activity in a center to support the workshops, mobility, and project engineering to search for funding. And Carlos is very deeply involved in barcoding and wants to give a big flavor of barcoding to that center. So that's quite promising. In terms of projects, so I start actually with a project that is led by Carlos Lopez-Varamonde from the INRA in Orléans. That's the CLIM3 project that is looking at the impact of the changes, the climatic changes, and the management of forest, mountain forest, on the diversity, on freshwater and insect diversity. And so that's one of the big projects at the moment that is building library for saproxylic beetles, and to a lesser extent for freshwater invertebrates and soil microinvertebrates, and it's involving 4 different countries: France, China, Germany, and Italy. A second project has just been recently funded by, so it's led by Stéphane Boyer from the institutes of research on the biology of insects. It's again in the central region, so there's a lot happening in that part of France at the University of Tours. And it's a project that focuses on food webs and understand how these food webs can be maybe also like managed and modified to improve the management of agroecosystems. And it's focusing on predator-prey, parasitoid host, and pollinator network. It's a pretty large project, like more than 500,000 euros, but in terms of barcoding library, it's relatively limited. So it's focusing on a limited number of species and using both meta-barcoding and barcoding. A third project that I am developing with Delphine J from the laboratory, the molecular laboratory platform at the Natural History Museum. It's actually a project where we want to invite researchers at the museum, other taxonomists and researchers, to submit samples to the lab for barcoding and to process them with NGS. So we are asking for funds at the museum now. We will get the reply at the end of the year, but that's something that I think we'll do anyway. So it's not a big funding; it's 15,000 euros, and we want to invite researchers to provide plates. They cover the cost of the extraction and PCR from their own project, and we cover the cost of sequencing. We buy the primers, the index primers that are rather expensive for the dual indexing with NGS. So that's what we are developing. There's a pilot project that is running right now on this with the same approach at the museum. It's based on the protocol that you published with Shadi a few years ago. And a fourth project that I added, it's not generating much barcode in terms of library, even though for one of the projects we are doing a bit of barcoding. But these two Actias and Sphinx projects, they are looking at my two pet groups, Saturnids and Sphinxids, and from the perspective of other discipline. And so these projects are developing tools, and the Actias project is a synthesis project. So it's meetings where we brainstorm about how we can develop pipelines, tools for using DNA barcodes to answer over biological and ecological questions. So one of them is like how to combine the barcodes with genomic data for diversification analysis, so in evolutionary biology, but also in macroecology, or we can use the barcode data to map the diversity, to model the distribution of diversity on the plant. And finally, for the SWOT analysis, of course, France has a large natural history collections. The museum has huge collections, but there's other public and a lot of private collections also in France. There are many type specimens that are very valuable when you build libraries for associating names to species. There's lots of taxonomic expertise and a very active community of taxonomists. A number of them are already involved in barcoding. In terms of facilities, there's one big facility, the Genoscope for sequencing, and then a lot of small facilities, small to medium-sized laboratory that can actually build NGS libraries or process like sample through extraction and regular PCR. Of course, it's in Europe, it's a highly diverse country. We have mountains, we have all the Mediterranean portal that is quite rich, and there's 40% of the European flora is represented in France. But in terms of diversity, most of that diversity is actually in the overseas territories. And I found these numbers that 98% of the French vertebrate species are actually in the French overseas territory. So in 20%, 22% of the surface of all France and French territories, that's where the richness is. In terms of weaknesses, of course, again, the difficulty to find funding for biodiversity research. We're all facing that, and I guess that's all why we're here, also to group efforts and get more leverage for changing that. And there's no national network, and the community is rather scattered in the country. Threats, that's that thing that we already mentioned, that all the regulation that about the Nagoya Protocol that can make our life more complicated to access genetic resources and to use them even just for producing DNA barcodes and building these libraries. This is actually a difficulty we have here for collecting in France or the French overseas territories to get all the permits for using them for barcoding. And on the opportunity side, of course, these overseas territories can be potentially associate members to iBOL and could contribute. There are like local research centers and institutions that are installed in these countries. Well, the territories, different sort of administrative level for these French territories. And also France has close relationships with French-speaking countries, in particular in Central Africa, with Gabon, for instance, and a few other countries, Madagascar, where we can maybe help and be instrumental in getting these countries involved in iBOL. And this is it. Thank you.